Here is a question that might seem simple. When you learn that a rule has changed, say, the exit at work has moved, what part of your brain is actually doing the rewriting, not just memorizing the new route, but actively replacing an old complex rule with a new one?
I'd probably guess some memory region, like the hippocampus.
A natural guess, but today's paper points to something far more local and surprising, a tiny tuft of branches at the very tip of a neuron in your motor cortex. When researchers blocked that tuft, animals could still perform every task they had already learned, but they could no longer relearn a complex rule. Welcome to OSCI Pod. Today, we are talking about dendrites as active decision makers.
Okay, dendrites. Those are the branching arms that receive incoming signals to a neuron, right?
Exactly, and for a long time the standard picture treated them like passive cables. Signals arrive, they add up at the cell body, and if the total is large enough, the neuron fires. Simple summation.
Like a voting system where each input gets one vote, and if enough votes pile up, the neuron shouts, yes.
That's a great analogy, but those branches turn out to be far from simple wires. Pyramidal neurons, the main excitatory cells of the cerebral cortex, have two structurally distinct compartments. The basal dendrites, sitting close to the cell body, receive specific feature-level signals, such as which direction a whisker was touched. Then there is the apical tuft, a cluster of fine branches at the far upper tip of the neuron that reaches into the outermost layer of the cortex.
And the tuft gets different information from the basal part.
Quite different. The tuft receives what the authors describe as contextual information, signals about what the animal is currently trying to do, what rules are in play, what broader situation it finds itself in. So the same neuron is simultaneously listening to two conversations, one about the immediate sensory facts, one about the larger context.
That's already a much richer picture than simple summation.
And here is what makes the tuft especially interesting. It can generate its own powerful electrical events called calcium spikes, surges of calcium ions that flood the dendritic branches. These are not passive echoes of the cell body. They are local amplifications that appear to play a role in how the neuron updates its connections.
So what was still unknown before this study?
The key open question was whether these tuft calcium events in the frontal motor cortex are specifically required for flexible learning, the cognitively demanding kind where you have to abandon a complex rule and then relearn it. Prior work had explored dendritic activity mostly in sensory areas or had looked at already learned behavior. Nobody had directly shown what happens to relearning if you silence just the apical tuft while leaving the rest of the neuron intact.
So what is the central idea the paper is testing?
The core proposal is that the apical tuft dendrites of a specific class of neurons in the motor cortex, called layer 5B pyramidal neurons, meaning they reside in one of the deeper layers of the cortex, serve as a critical computational resource for updating complex behavioral rules. Not for executing a behavior you already know, but specifically for the cognitive work of relearning something hard.
And how do you isolate just the tuft? It seems nearly impossible to target one part of a living neuron.
Sharp question. The researchers used a beautifully specific biological tool. There is a class of inhibitory neurons called NDNF interneurons. NDNF stands for neuron-derived neurotrophic factor, but what matters functionally is where they live and what they connect to. These neurons reside exclusively in the outermost cortical layer, called layer 1, and their axons reach directly down onto the apical tufts of the pyramidal neurons below. They act like a dedicated volume knob for the tuft and nothing else.
So if you activate the NDNF interneurons, you turn down the tuft specifically.
Precisely, NDNF neurons release the brain's main inhibitory messenger, called GABA, in a diffuse manner across a wide area. This activates a specific receptor type called GABA-B on the tuft dendrites, which suppresses calcium channels there. The result is that the large, coordinated calcium events throughout the dendritic tree are essentially switched off, while the tiny, local calcium signals at individual synaptic spines are largely preserved.
Wait, so it is like shutting off a broadcast signal while leaving individual whispers intact.
That is a perfect image. The whispers, local synaptic inputs at each little spine, still happen, but the coordinated, whole-tree calcium broadcast that reflects burst firing is eliminated. This dissociation turned out to be crucial for interpreting the results.
All right, how did they test whether any of this matters for learning?
They designed a rule-switching task for mice. First, each mouse learned a complex rule. The authors call it Rule A. A mild air puff was delivered to either the left or right side of the animal's face, and the mouse had to lick toward whichever side was puffed in order to get a reward. This required a context-sensitive, bidirectional decision on every trial.
That does sound demanding. Left puff lick left, right puff lick right.
It took the mice roughly 2 weeks to master it. Then the rule switched to Rule B, much simpler. Regardless of which side was puffed, the mouse should always lick left. No context needed, just always go left. The mice picked this up quickly.
And then they had to switch back to the hard rule.
Exactly, that return to Rule A, which the authors label Rule A', is the critical test. It is not just learning something new, it is reinstating a cognitively demanding bidirectional rule after a period of simpler behavior. This is where flexible learning is really being measured.
And the NDNF manipulation was applied during this relearning.
Each mouse went through the whole paradigm twice, once under normal conditions, once with NDNF interneurons being optogenetically activated during a specific 3-second window after each trial, when the animal was assessing the outcome. The researchers reasoned that learning requires a feedback signal, reward, or error, and that is precisely when the dendritic plasticity mechanism should be operating.
So they targeted the moment of learning, not the moment of performing.
Exactly. To see inside the dendrites, they used 2-photon calcium imaging, a technique that uses a focused infrared laser to detect fluorescent light from calcium sensors expressed in specific neurons. This allowed them to watch calcium activity in individual dendritic branches and even in individual synaptic spines, those tiny protrusions on the dendrite, while the animal was actively performing the task.
Watching individual synapses in a moving, behaving animal, that is remarkable.
They also recorded electrical activity with NeuroPixels probes, ultra-thin silicon devices that can monitor hundreds of neurons simultaneously to compare what was happening at the cell body versus the tough dendrite. And for the synaptic input side, they used a separate fluorescent sensor that lights up specifically when the excitatory messenger glutamate is released, allowing them to map which synapses were active and where they sat on the dendritic branch.
What did they find?
The most striking result, activating the NDNF interneurons, silencing the tuft, had no effect whatsoever on Rule A performance once it was already learned. The mice licked correctly, just as before. It also had no effect on learning the simpler Rule B.
So the tuft is not needed for doing something you already know, or for learning something easy.
When it came to switching back from Rule B to the complex Rule A', activating the NDNF interneurons dramatically slowed relearning. The mice made significantly more transition errors, trials where they defaulted to the old rule even after the new rule had been introduced. And notably, this impairment persisted into the following day after the optogenetic stimulation had been turned off, confirming that the manipulation had affected the learning itself, rather than just momentarily disrupting performance.
So silencing the tuft left everything intact, except the capacity to relearn something complex.
Now look at what was happening inside the dendrites. When NDNF interneurons were activated, global calcium activity in the dendritic shafts dropped by roughly 77%, but the firing rate of the cell body only fell by about 23%. Those are very different magnitudes, and they point to the calcium suppression being primarily a dendritic phenomenon rather than simply reflecting reduced cell firing.
The dendrite was hit much harder than the cell body. That really does show the two compartments behaving separately.
And as expected from the mechanism, the local calcium signals at individual spines were barely affected. The whispers continued, only the broadcast was silenced. There was also a specific reduction in burst firing, those rapid trains of multiple action potentials, and the effect on bursting was larger than the effect on ordinary single spike firing. This fits with the idea that bursts are the physiological correlate of the global calcium events in the tuft.
What about the synaptic inputs to the tuft? What were they doing?
This is where the clustering finding comes in. Under Rule A, nearby synapses on the same dendritic branch tended to fire together in correlated patterns, what the field calls functional clustering. Under Rule B, this clustering disappeared. The inputs were still arriving, but they acted independently rather than as coordinated groups.
Wait, so the complexity of the task physically reorganizes how inputs coordinate on the same dendritic branch.
That is what the data suggest. And this clustering was specific to synapses encoding choice information, which side the animal decided to lick. Synapses encoding the simpler sensory instruction were distributed more randomly across different branches rather than clustered together.
How do all these pieces fit into a single mechanism?
Here is the picture the authors propose. During a complex rule, thalamic inputs from motor planning regions and sensory inputs from primary sensory cortex converge on the apical tuft. Their convergence generates large, synchronized calcium events that propagate through the entire dendritic tree. These events are tied to burst firing. The authors suggest that these bursts drive a form of synaptic strengthening specifically. They enhance cooperation between nearby synapses that carry consistent, rule-relevant information. Over time, this cooperation becomes the functional clustering we observed.
So clustering is not just a side effect, it is the memory trace being written.
That is the proposal. Now here is the really elegant piece about the NDNF interneurons. The researchers found that the NDNF neurons themselves reduce their activity specifically when the animal makes errors related to learning, both during rule switches and during initial learning of a new association. But crucially, their activity does not decrease during spontaneous errors in already learned behavior.
So the NDNF neurons can distinguish between "I made a careless mistake" and "I am actually in the middle of learning something new."
That is the implication. When a learning-related error occurs, the NDNF interneurons go quiet. Their inhibitory hold on the tuft is released. This disinhibition opens the gate for large calcium events to flood the dendritic tree, enabling the plasticity that updates the rule. When behavior is stable and well-learned, the NDNF neurons stay active, the tuft is suppressed, and the existing representations are protected from being overwritten.
It is a kind of era-triggered plasticity gate. The neuron only allows rewriting when it is actually needed.
And the upstream signal that may close the gate during learning is likely acetylcholine, the neuromodulator that the brain releases during unexpected or significant events. The authors observed that pupil diameter, a proxy for acetylcholine activity, increased during error trials. The proposed chain is: learning error leads to acetylcholine release, which silences NDNF interneurons, which unleashes calcium in the tuft, which drives the synaptic changes that update the rule. The whole circuit is organized to protect stable behavior while selectively enabling rewriting when the world demands it.
That explains a puzzle I had: how the brain stays stable most of the time, but can still reorganize rapidly when it needs to.
That is precisely the contribution. The two-compartment architecture of pyramidal neurons, where the tuft receives context and the basal dendrites receive features, combined with this irrigated inhibitory control, gives the cortex the ability to be simultaneously stable and flexible.
So the big picture is that the apical tuft dendrites are not just passive listeners. They are active computational units that specifically enable the brain to update complex rules while staying out of the way during ordinary behavior.
And the NDNF interneurons sitting in that outermost cortical layer are the gatekeepers, normally keeping the tuft suppressed, but stepping aside precisely when learning-related errors signal that a rule needs to change.
If I had to put today's surprise in one sentence, the gate to flexible intelligence is opened by the right kind of mistake, and it lives at the tip of a dendrite.
That is exactly it. Today's episode was based on tuft dendrites in frontal motor cortex enable flexible learning by Eduardo Maristini de las Casas and colleagues, published in Science in May 2026. Thank you for listening to OSCI Pod.
See you next time.